Well, good afternoon, everyone. Welcome to our CCOVI Lecture Series today. I'm going to be introducing myself as the speaker. I just realized that all the introductions I've been doing throughout the term, I keep forgetting to tell you who I am. So as the speaker today, I'm Dr. Debbie Inglis. I'm the director of the Climate, Enology and Viticulture Institute here at Brock University, and I'm also one of the researchers affiliated with the institute here. I'm a faculty member in the Department of Biological Sciences and have research programs in various areas of enology research in terms of yeast fermentation, stress responses, ice wine production that is now branched into Appassimento wine production, which is what I'm going to speak about to you today. In terms of my background, I received my PhD in biochemistry from McMaster University, just down the road here in Hamilton, and joined CCOVI in 1999. Actually joined Brock University, came back to academia after 3 years in industry, but specifically I came back to Brock to be part of the Climate, Enology and Viticulture Institute that was just opening up at the time. Since that time, I've been an active member in my Department of Biological Sciences and now took over as director of CCOVI in 2008 and still serve in that role today. So please sit back and relax as I speak to you today about Appassimento wines and the role that we're playing with this research program to help the industry develop a signature style for Ontario. So in terms of why we're doing this project, I just wanted to get into a bit of background into why this became an issue for us to look at in a research project for us to look at. And it falls under the topic of climate change and what that means for us here in the region. With climate change upon us, we actually have quite a bit of volatility in our weather. We have extreme temperature highs during the growing season that can affect fruit quality and vine survival. We have extreme cold winters, weather in the winter, and we sometimes have an excessive amount of rain or lack of rain through the growing season that can impact the quality of our fruit. With that climate change, how is that impacting growing season from year to year? Well, if you just take a snapshot of what's been happening with our industry in the last decade, you can see from this table we've got quite a bit of difference in weather throughout the growing season. In 2005 and 2003, actually through the dormant winter months, these were known as our short crop years. It became so cold during this time, we actually got damage to our vines and reduced a lot of the production in those years. In 2007, it was a very dry drought year for us. We had no problem ripening our reds, but it was actually a little bit too hot for our white varieties. In 2008 and 2009, they were our very wet, cool years, so the complete opposite from what we saw in 2007. 2010, we had marked as just a great vintage. Everything seemed to come at the right time, the rain, the sunshine. It was a great harvest. 2011, back to a bit of a late cool spring, wet fall, some disease pressures. And then now our newest vintage, 2012, we now think is the next best vintage. Everything again seemed to come at the right time. So why I draw your attention to this is that our winemakers on a yearly basis have to deal with all of these extremes that are occurring within the growing season and the dormant season to ensure that year in and year out we're producing top quality wines for our industry. So this allowed us as the research community to really look at taking a proactive approach in partnership with our industry to look at ways to mitigate the impact of climate change by focusing on both viticulture issues as well as enology issues so that we're prepared and can figure out what these impacts are going to mean to us and overcome them. In terms of viticulture and climate change, we have definitely an impact on the wine regions, so all the sub-appellations that occur within Ontario. Each of those have unique microclimates that can be affected by, that can be affected by the temperature. In terms of enology and climate change, what this means is that it might open up new varieties that we can develop for Ontario that actually takes advantage of the climatic issues that we're trying to deal with. So it leaves open the opportunity to develop some new wine styles, such as Appassimento wines, which is what I'm going to focus in on today. In terms of the funding for this program, I would be remiss if I didn't talk about where the funding comes from for such an immense program that I'm going to talk about today. We were fortunate to receive funding from the provincial government through the Ontario Research Fund Research Excellence Program. This is a partnership being led by CCOVI here at Brock, but it also involves our research partners at Niagara College, University of Guelph, and Vineland Research and Innovation Centre. We have a number of industry partners, such as the Grape Growers of Ontario, Ontario Grape and Wine Researching, VQA Ontario, who's very excited about this new wine style that's developing for the region. We have wineries and grape growers on board. We have a greenhouse operation on board. We also have another federal funding agency, Agriculture and Agri-Food Canada. So overall, this climate change program in viticulture and enology is actually a program worth over $10 million, with $2.86 million coming directly from the province. So we're actually into year 3 of this particular project. Hi, Graham. One of our industry partners, 2 of our industry partners, 3 of our industry partners just walked into. No problem whatsoever. So I'm just started out giving a little bit of background about why we're interested in this project and where the funding is coming from. So again, in terms of our enological short-term strategies, we're really looking at adoption of methods and technologies that will mitigate some of these risk factors and stabilize the wine industry from year to year. So how do we take advantage of cool, less optimal years when grapes may not fully ripen during that growing season? Well, when those sorts of seasons happen, we know we have higher levels of acidity in our grapes. So maybe we could put a little more focus on some of the wine styles, such as sparkling wine production, that actually require a high level of acidity in the grapes. What's another option? Well, we could look at ripening the fruit post-harvest. So not relying on Mother Nature to ripen the grapes, but look at some of the biochemical and microbial interactions that could occur on the fruit post-harvest and see if we can develop a unique wine style for Ontario based on this Appassimento technique. Thirdly, another part of our enological approach is reducing green characteristics in wines through reducing methoxypyrazines. You'll have to come back for another lecture on another date to hear about the exciting research we're doing in this area. So today I'm really just going to focus on the Appassimento portion of this very large research program. So traditionally, this method of ripening grapes post-harvest utilized techniques that allowed for a very slow progression and ripening of the fruit, somewhere between 60 and 100 days. Typically, this post-harvest ripening would occur at fairly low temperatures with lots of attention paid to the humidity levels that were present during that drying process. The results definitely depend on the surrounding air conditions that the fruit is exposed to. So temperature, relative humidity, and airflow. All of these factors will have an impact on how fast the grapes dry and a lot of the biochemical transformations that will occur within the fruit. Also, we need to be aware of the characteristics of the grape varieties that we're putting through this drying process. How thick are the skins? What sort of cuticular waxes are on these grapes? That may have an impact on how fast these grapes are drying through this process. Overall, with this approach, we're looking to further concentrate the flavors and the sugars that are present in these grapes, but also at the same time not concentrate things like acidity that also would just have the propensity to concentrate if we were just removing water from the fruit. So in terms of developing this wine style, the varietal characteristic can definitely have an impact on the overall wine style that results. So one of the things we had to figure out as an industry research group, what varieties did we want to focus on first and foremost for developing the style for Ontario. And keep in mind that the biochemical changes that are occurring in the grape are not only due to metabolic pathways that get activated and are still active in the grape, but there's also microbial interactions. There's all kinds of microflora that can be present on the surface of the grape. And under these post-harvest conditions, the microflora can also have a contributing impact on the overall flavor profile of the final wine. So in terms of the potential role for Appassimento wine, red wine production in Niagara, I just wanted to draw some comparisons to what's happening in the very well-known region in northern Italy for Appassimento or Amarone style wine production. It's definitely a proven production outcome using this method in producing Amarone style wines. So the question is, can we adapt this technique and develop a wine style for Ontario, and here specifically right now in Niagara? So we're looking at increasing sugars, reducing acids, and evolving unique flavors that are going to have a signature that immediately somebody will recognize that wine style. Oh, that's the Appassimento wine style from Ontario. Granted, yes, I know we need a better name for that, and I think we're working on that through the VQA Ontario, but we want these wines recognizable as a unique signature coming through for Ontario. With the Appassimento wine production, further ripening the grapes post-harvest, we're removing the grapes from all of the climatic risks associated out in the vineyard. So we're removing the grapes from risk from humidity and frost conditions that can occur out in the vineyard in our fall period, but also from wildlife degradation and further breakdown of the fruit. In terms of a proven market impact for Niagara, might it be comparable to what we've seen happen in Italy? So in terms of industry growth, Amarone, specifically just Amarone wines, 1.5 million bottles 12 years ago, and in 2012 that grew to 12.5 million bottles. Okay, so lots of room for growth here. If we also take a snapshot of just the breakdown of these wine styles from 2007 to 2010, in 2007 and in 2010, we're, upwards of 50, 55 million bottles. But if you look at the breakdown, the shift has gone from non-Appassimento style wines through to a much larger focus on Appassimento style wines. Why does this have an impact on the bottom line for the industry? Well, because these Appassimento wines, they target and command a higher dollar value in sales. So they can make a significant contribution.
What is that last stat? Is that an Ontario stat?
No. Yeah.
Total sales in LCBO, is that what you're saying?
Yeah, not LCBO. This is just sales figures from a report done mainly out of northern Italy.
It's production numbers.
Yes.
And I noticed that it includes Valpolicella and Ropasso.
So, yes.
So that's why we're going from 15.5 million to
31.
12.5
Yeah.
to 15 million.
Yes.
Because the rest is probably Ropasso.
Yes.
Okay.
Yes.
Okay.
So the point here is just trying to make that, there's a big shift in moving towards, this Appassimento higher end wine product made from grapes post-harvest, and away from just non-Appassimento table wine production. Okay? In a relatively short period of time. Okay? So this wine style is really growing in popularity. So in terms of our project, what are we looking at? So we started out looking to assist the industry in developing this wine style by looking at different ways to dry the grapes. So there are wineries that are already commercially here in Ontario producing this wine style. But wineries are sorry, there's a timing issue here on the slides. The wineries are using different techniques to actually dry the grapes. So our control method is to just leave the grapes on the vine and allow them to just naturally further ripen on vine. Okay, that's one of our controls. We're looking at traditional barn drying method, and we're utilizing the barn at Cave Spring Cellars for this technique. Drying the grapes in a greenhouse operation. We've had 2 greenhouse partners involved with this. Most recently, European planters have come on board for this technique. Using refurbished tobacco kilns. This has grown in a lot of popularity in terms of a quick way to dry the grapes. And a new technique being developed at a Vineland Research and Innovation Center is using a specialized drying chamber where the humidity and temperature can both be very tightly regulated. So for this project, we're looking to assisting the industry to develop this Appassimento wine style by comparing what happens with the fruit chemistry and the resultant wines if we dry the same grapes using all 5 of these different drying techniques. We're currently into year 3 of the study, and we started initially focusing on using Cabernet Franc. Okay. Cabernet Franc is the most dominant red variety planted in the region. Just based on the structure of the grape cluster and the grape skins, we thought that it would be a good variety to trial in these studies. And several of our partners were all industry partners, wineries, were already producing Appassimento style wines using this grape variety. So it was a great fit for us moving forward. In addition to following what's happening in the grapes throughout that drying process, we're also interested in looking at the microbiology and how the microbiology is changing on the surface of the fruit during the dry time and using those different techniques to dry the grapes. And of course, the study wouldn't be complete unless we analyzed the must in the wines both chemically as well as sensorially. So in terms of the methods that we're using, Pilotary Estates Winery, and Jamie Slingerland is in the audience today, was kind enough to supply all of the Cabernet Franc grapes for the study. We separate the grapes at harvest into the different treatments, and they're delivered to the different drying facilities. And we keep one of the lots of grapes hanging on the vine so that we can use that as one of our controls. And another control is just to harvest the grapes and produce wine at normal fall harvest time so that we can see what the wines would be like if they didn't go through any further post-harvest drying. We target the fruit at harvest at 23 bricks. So these are fairly ripe grapes to begin with. They're not overcropped. They're cared for properly out in the vineyard. They achieve that 23 bricks at normal fall harvest time. The target for further drying the grapes are at both 26 bricks and at 28 bricks. The 26 bricks, we've, in consultation with our industry partners, they felt 26 bricks would be probably the more commercially applicable level to target the grapes to dry. That would produce a wine, upwards of about 14% alcohol. But being researchers, we also wanted to push the limits a little bit and see what would happen if we actually dried those grapes further up to 28 degrees bricks. And with the 28 degrees bricks, that can produce a wine upwards of about 15.5% alcohol. So these are fairly big wines that were generating full-bodied, lots of alcohol present. Yes?
What year was this particular particular study?
So we've done this study for 3 years now, but I'm going to be presenting you data from the 2012 harvest as well as the 2011 harvest. The 2010 harvest was more of our trial year, just to get our techniques on board and figure out exactly the best way to facilitate the research project. So all of the wines are fermented using the same protocol. So we start with the same grapes. They're split up. They go to the different drying conditions. Once they reach the target Brix, we ferment the grapes using the same protocol. We use EC118, which is a yeast recommended to us by L'Allemande Italia, which is located in Verona, Italy, where, you know, the home of Amarone style wines. And we do all our fermentations in triplicate so that we have a robust data set. Yes, you had a question.
Maybe I missed that, but which one of those drying techniques have you tried? Some of them you lifted on the vine, that's okay, but the ones
We've yes, all of them actually. So this program, we compare all of these drying techniques.
And you compare each drying technique system?
Yes. Okay. So the vast data set that we're going to go through will contain data from all of those techniques. Yes?
The grape on the chart, donated grapes for each of the 3 years?
Yes.
Same field.
Same field. So we've got consistency. So our harvest date doesn't always end up being the same year. So 2011, our harvest date I think was October 17th, whereas 2012 it was an earlier year. Our harvest date was October 4th. But we're targeting the same Brix level in the grapes for this. So I wanted to start with the data analysis, and hopefully I'm not going to scare you off with some of these crazy graphs. But it's important for each of these techniques to take a look at what the impact is of humidity, okay, in blue, sorry, and temperature that's present in red on these graphs. So this is the data from the on-vine treatment. And you can see there's lots of variability in humidity, there's lots of variability in temperature. And you can also see in 2012, we had quite a few days during this drying period where the temperature dropped below 0 degrees Celsius. So these grapes were actually going through some freeze-thaw cycles as well. In 2011, similarly, I've just put these graphs in black so you can tell the difference. But we've got lots of variability in humidity. We've got lots of variability in temperature. So again, these are difficult conditions to just naturally hang the grapes out in the vineyard to achieve our 26 and our 28 Brix. We do achieve that, and we leave the grapes out there until we do. But the climatic conditions are quite harsh for these grapes to be able to not undergo some breakdown during that period when they're exposed to the elements in this manner. So there's higher risk associated with just hanging the fruit out on the vine. So it's not that we can't achieve these Brix levels, but there's definitely much higher risk for the winery to leave the fruit out here, given the fact that our fall harvest conditions are usually quite variable in humidity as well as temperature. When we take a look at the barn condition, so within the barn, we're looking to target sort of a mid to long-term duration of drying. So again, depending on the external climatic conditions, it may take a month up to 2 months to reach our target Brix level. So in terms of what's happening here in the barn, if we were to compare the humidity and temperature to what was happening on that previous graph, from the external conditions out in the vineyard, we would see there's quite a tight correlation between humidity and temperature in the barn compared to what's happening outside. So basically what happens outside during the fall harvest is going to be mimicked within the barn setting because we're not controlling the temperature or the humidity in that manner within the barn. It's exposed to what's happening externally to that building. Now, what is protected is the fruit from wildlife predation and some of those extremes. So you'll notice we don't get quite down to the cold below 0 degree temperatures in the barn. But as the fall months get cooler in temperature, so does the temperature within the barn.
There's no heating in the barn at all?
There's no heating in this barn. Okay, it's done in a very traditional method. The only thing that is present in this barn are circulating fans to help with the air circulation. Okay. And again, I put the 2011 data up here as well, just to show the comparison with 2011 compared to 2012. So again, our dry time in that 1 month to 2 month time period. On vine, our dry time was that 2 month, maybe 2.5 months to reach our target levels. In terms of the greenhouse, this has been an interesting experiment to figure out the best way to use the greenhouse operation. Now, in 2011, as a research group, we thought of a way to look at drying the grapes in the greenhouse, but trying to really control the temperature and humidity within that greenhouse setting. So as the grape bins were piled up in the greenhouse, they were covered with a tarp material. And the idea here was that we would better be able to control fluctuations in temperature. What we weren't counting on underneath the tarp was that it was also actually building up humidity. Even though there were circulating fans, it wasn't enough. And so in the early stages in 2011, in the greenhouse under the tarp, we built up too much humidity, and it was still fairly warm. These temperatures are, getting up over 20 degrees Celsius, and the humidity is over 90% relative humidity. Those are really good conditions for botrytis to build up. And unfortunately, that's what happened with this trial in 2011. So midway through the trial, I had to make a decision, being the lead investigator on this, and we decided to remove the tarps from the grapes. We got rid of the gray mold fruit, botrytis, but it was in the gray mold form, fuzzy gray fruit. Okay. So we discarded that fruit, cleaned up the remaining grapes that were in the bins, and then just continued with using the greenhouse. Now, by this time, we were getting into well into November. The temperatures were starting to get quite cool, so the greenhouse operator turned on the heat to try to keep our temperature a little bit warmer, because we were really targeting this technique to be a mid-dry technique. It would take 2 to 3 weeks to dry the grapes. So in 2011, we were off a little bit on our target. The dry time was about 29 days, so almost a month. And we realized, okay, with this trial, putting the tarp over the fruit, that didn't really work for us. But that's why we do research, so that we take the risk as researchers and share that information with you so that you don't make the same mistakes when you go out to do this on a commercial scale. In 2012, we decided to go with a different setup in the greenhouse. So we had a greenhouse operation, European planters. They had tables within their greenhouses that we could put our drying trays on. There was lots of air circulation around the grapes. The grapes went in. They only took between 12 days and 18 days to reach the 26 and the 28 bricks. You can see here for the red scale, this is temperature. So we were holding the temperature for the most part, around that 15 to 20 degree temperature range, which worked out well for this. And the humidity levels, for the most part, other than the first couple days, were held below 90% relative humidity. So we had very little breakdown, very little botrytis that developed in this fruit. We were able to show that, yes, the greenhouse is a viable operation for this midterm time, a couple of weeks to reach our target levels. Also, with going in the greenhouse, of course, the fruit is protected from the external environment. So again, it's somewhat like the barn, but even a faster turnaround time that we got for drying the fruit. Our fourth method was the kiln method. Now the kiln method, you'll notice here on the scale, it's a much shorter time frame. We're talking about days here with the kiln. So the kiln was targeted to really dry the fruit within a couple of days. In 2011, I'll start here. It took about 3 days for us to reach our first target. You can see our temperature was up around 35 degrees Celsius. We removed the grapes from the kiln, opened up the kiln doors, temperature dropped right away, put the fruit back in, built the temperature back up, dried them for a couple more days. We got up to 28 brix. So this worked out quite well, and you can see the humidity graphs basically are the inversion of what we're getting for the temperature. So we do have air circulation within the kiln that's occurring. We are using heat in this experimental design to dry the fruit, and it's a fairly rapid drying process. In 2012, we used the same kiln again. Started out, we weren't quite as calibrated on our temperature, so it only took a day for the grapes to go from 23 brix up to 26 brix because we were having that temperature in between the 35 to 40 degrees Celsius. After we took the first batch of grapes out, we got the temperature dialed in, held it at 30 degrees Celsius, which is what our target was, and took 3 more days to dry the fruit up to 28 degrees brix.
Is it easy to calibrate the temperature in a kiln?
It is. There was it wasn't due to the calibration that the temperature we mistargeted the temperature. It was a misunderstanding of somebody at the operation, and they dry their fruit at a warmer temperature. Didn't realize that we were doing an experiment in the kiln. So once we got the kiln at the temperature, everybody was aware. It was very stable. We were right on that 30 degrees Celsius. Okay. So again, our shortest time period. Yes, Jamie.
Was there any reason to pick 30 Celsius and not pick 25? Was there any reasoning that it had? And I realized some people had the concept of going higher.
Yeah.
But potentially there could be a much lower temperature.
It could, and I think what we were trying to target with all of the techniques was looking at different dry times. So we wanted the kiln to be the most rapid dry time. So with that, we wanted to utilize some heat, control the humidity that way. It's a forced air system. We targeted 30 degrees Celsius just through discussions with our industry group. 25 would be basically room temperature, maybe a little bit of heat because we are getting into the fall months. 40 we thought might be too rapid, which yes, in the end was too rapid when we went up that high. So 30 was just our educated guess. Let's start with 30 degrees Celsius. Yes?
What's the temperature
in Italy, they traditionally use the barns, okay, for drying the grapes. So they're exposed to whatever the external temperatures are. So much like what we see with our barn here, that we've got much more variability within the temperature and humidity, because in Italy, the barns that are used for this, they open up doors, they allow the temperature, the airflow to come in from outside. They don't get quite as cold a temperatures as what we get, into the late November, December months. But again, they're looking at a slower drying process. Yes.
Massey has massive facilities now that
Yes.
complete climate control, so
Yes.
it'd be probably interesting to pick up data from them to see what their temperatures
Yes.
are, because
When they share that. Yes.
Yeah.
Yes. Yes, I have been to the Massey facility, and there are some newer technologies coming on board. If you see any of their facilities, they very tightly control, monitor, I should say. They very tightly monitor their conditions. The movement to temperature-controlled and humidity-controlled chambers is an issue in northern Italy. It's taking away from the traditional style of producing Amarone. So there's lots of discussion right now, and I'll talk to you a little bit about that as we continue with the talk, in terms of artificially controlling the conditions for drying and what the benefits and repercussions are to that versus going the more traditional way, which is just exposing the fruit in some kind of covered, barn facility, but still exposing the fruit to the climatic conditions externally. All right, and getting to our last treatment that we trialed in 2011, this is the temperature and humidity controlled chamber from Vineland Research and Innovation Center. So the idea here is that using dehumidification and temperature control, you can really precisely control how long you want to expose your fruit for before they reach the target temperatures. So you can see here in this graph, just visually looking at it, they're much tighter control over humidity and temperature. Okay, so with the setup that they have, they can very tightly control this and hence control how long it takes to dry the fruit. And in this particular experiment, we were trying to target to dry the fruit, between about a month and a half. So it ended up being 44 days to reach the 28 bricks. A little shorter for the 26 bricks. Okay, so again, in this technique, the fruit is also protected from the elements, but there's the added benefit or layer that you can really precisely control humidity and temperature, and through this, there's mathematical equations that the research specialists, Bernard Goyette, can work out to really dial in how long you want to dry the grapes for. Yeah.
Dry chamber, you mean the one in the barn?
No, this is a specially constructed drying chamber. Well, it was a room that they specially constructed to control the humidity and the temperature within the facility.
That's different than the first three that we went through.
Yes, so if, sorry, I'll just backtrack on my slides here. So this is the drying chamber, this is the barn, this is the kiln, this is the greenhouse, this is on vine. So they're each separate facilities that were
Debbie, one of the big
being used.
differences in the drying chamber that's not evident here is, whereas most of these facilities, including the Massey, have fans blowing on, here we use a suction to suck air through that whole stack of grapes to get more even distribution.
Yes. Whoop, sorry, wrong way. So I think you can see from that compared to the other techniques, there's quite tight temperature and humidity control using that system. Okay, so on to some of the experimental results on the fruit. In terms of reaching our target bricks level, again, for each of these graphs, I'm going to have data from 2011, and I'm going to also have data from 2012. They basically tell the same story. And the story here is that each of the methods, we reach our target bricks, 26 and 28 bricks. So since this is a time course, we're going up to 28 here. We're able to reach this with each of the techniques, but each technique takes a different amount of time on our bottom scale to reach that target level. So we're using different technologies to dry the fruit, and will that difference in time and the climatic conditions that the fruit are exposed to, will that result in different flavor developments in the fruit and slightly different styles of wine that result because of this? Just to summarize what those values are in terms of harvest date, I'm going to stand here because there's some kind of timing that's gone on to these slides, and I have to turn them off. Otherwise, we keep going through the additional slides. So in terms of 26 bricks, you can see that in 2011, it took us about 30 days to reach that bricks, whereas in 2012, it took us 13 more days to reach that 26 bricks out on the vine. Similarly, to reach 28 bricks, took a little bit longer to do that in 2012. A little more erratic weather in 2012, and you remember back when we looked at the temperature and humidity values for 2012, we had quite a few days where we were going down below 0 degrees Celsius. It's a little bit cooler that fall, and hence it took a little bit longer to reach our target bricks. For the kiln, in 2011, 3 days to reach that 26 bricks. 2012, we had that higher temperature, it only took us a day. The barn, in 2011, it took us 22 days to reach that 26 bricks. In the following year, it only took us just over 2 weeks, okay, to target that level. For the greenhouse, again, 2011, not really representative of the greenhouse because we did that tarp experiment. But if you look at 2012, it took us less than 2 weeks to reach that 26 bricks. And for the drying chamber, again, where they were purposefully trying to draw out the length of time for drying, it took 22 days to reach that 26 bricks. In terms of weight loss of the berries, basically we would go through and take a sampling of 15 berries on each sampling day and weigh the berries and look at what sort of weight loss we were getting. So overall, in 2012, we saw about an 18% drop in berry weight, whereas in 2011, we had about an 11% loss in berry weight. I'm going to come back to these numbers a little bit later on near the end of the presentation, where I'm looking at comparing the cost associated with bringing the fruit from those different drying conditions to make the same volume of must for fermentation, and this will, the difference in weight loss from the different techniques will have a little bit of an impact on the financial side of things. So in terms of other measurements that we did in the fruit and in the wines, what were some of the things we were concerned about or looking for these Appassimento wines? With drying the grapes post-harvest in these drying facilities, one of the things we need to be concerned about are the increase in production of some oxidation compounds in the grapes. Compounds like acetic acid, acetaldehyde, ethyl acetate, that may start to build up in the fruit even prior to fermenting, okay, just because they're further drying post-harvest. We also wanted to keep a very close eye on botrytis. How much botrytis infection were we seeing in the fruit, and what sort of decay were we getting under the different conditions? Because you can see in all of these different drying techniques, we have different temperature and humidity conditions that resulted. So I'm going to walk you through some of the data and highlight, especially for the industry players, what some of the areas you need to keep a closer eye on depending on drying technique that you're using. So in terms of acetic acid, what we have found both in 2011 and in 2012 is that there definitely, for the kiln method, the fastest drying method, there tends to be an increase in production of acetic acid over the period of time to reach that target bricks. Okay. And these endpoints that I'm showing here for acetic acid versus time is the endpoint to reach 28 degrees bricks. Now, it's not that these levels of acetic acid are excessively high, okay? So even in 2012, the acetic acid in the berries got up to around 0.13 grams per liter. But I draw your attention to this because there is a propensity with the faster drying technique, and I think it's probably related to temperature and the microflora on the berry. We tend to get higher production of acetic acid. In 2011, the level produced was actually even lower. It was just over 0.09 grams per liter. Okay, so again, not getting to really high levels. Just to put it into perspective for everybody, typically grapes are rejected at the scale house if the volatile acidity or acetic acid level got up above 0.24 grams per liter. So we're still below that level, but it's just something that was quite striking when we compare all the different ways to dry the grapes compared to the kiln For some reason, the kiln is producing higher levels of acetic acid in the fruit.
Hard to see that peak there.
Sorry.
That number?
This one right here, it's 0.4, 6, 8, 10, or 0.1. So this would be about 0.09 grams per liter. Okay. So again, what we're investigating with one of the students in the lab, Christine Huber, who's in the audience today, we're actually looking at the bacterial microflora on the surface of these grapes, because what we think is happening at the warmer temperatures, there may be just a bit of an increase in some bacteria that's leading to these higher levels of acetic acid that aren't building up to quite the same degree in the other drying techniques. So again, not that it's at that high of a level, but it's just something to keep an eye on depending on how long you're going to be drying your fruit for in the temperature. Similarly, acetaldehyde is another one of those oxidation compounds. And similarly, we find that both the kiln as well as on vine tends to produce higher levels of acetaldehyde. Higher levels compared to the other methods. These levels of acetaldehyde are still quite low, okay? It's only 0.12 milligrams per liter, 12 milligrams per liter. So we don't even get into the sensory detection of this probably for another 10-fold. But again, the propensity here is that with this technique, we're getting a little more of this compound being produced. Malic acid, one of the compounds in the fruit that we want to see what happens during the drying process. Malic acid is one of the dominant grape acids in the fruit, and we certainly don't want to further concentrate that acid as we're concentrating the fruit, because we are concentrating the fruit. The fruit is losing water. What we find with malic acid, when it doesn't change, okay, when the level is fairly constant throughout this sampling drying period, keep in mind that the fruit is actually concentrating. Okay, it's going from 23 bricks up to 28 bricks. So in actuality, even though when we measure the concentration in the fruit, it isn't changing, it's actually dropping in concentration because we're further losing water and concentrating all the components within the grape. So if we weren't losing any malic acid, what we would expect to see over a time course is just a slight increase occurring. As the fruit becomes more concentrated, we're losing water, everything would become more concentrated. But what we see is that the malic acid either stays fairly constant in concentration or actually starts to drop off. So what this means is over that drying period of time, we're actually losing malic acid. Some of it is getting metabolized by probably within the grape itself through this drying process. So that's a good thing because we wouldn't want to start with 2 grams per liter of malic acid and concentrate it up to 4 grams per liter during this drying process. So what we're finding in 2012, we actually started with a lower level, somewhere between 0.15 and 0.18 grams per liter malic acid, and basically ended up with that concentration in our fruit once it dried prior to taking it through the fermentation. 2011 was a little bit of a cooler year compared to 2012, and you can see the malic acid started up a little bit higher. It's around 2.4 grams per liter here. We also saw some increases in glycerol during this drying period, and I only have data for this so far for 2011. We're still working on the 2012 data. You can imagine there's a lot of samples for us to work through with the size of this project. So not so surprising that glycerol increased throughout the drying period. It's what we would expect, some increase in glycerol as the fruit concentrates, but also there may be some contribution from some botrytis activity. Similarly, gluconic acid also increased 2 to 5-fold through the drying period. So again, what we would expect to see. In terms of ethanol concentrations in the wine, we can see that in our starting juice, we have very little, almost non-detectable levels of ethanol for any of our treatments. But when we take our fruit at 26 bricks and ferment it, our control wine is coming in at approximately 12.5, maybe almost 13% alcohol from that 23 Brix juice. When we further concentrate the fruit up to 26 Brix, we see for most of the treatments we are getting a higher alcohol level, which is what we would expect. Why we got a lower alcohol level in the on vine probably has more to do with just some of the breakdown that was occurring in the fruit. During fermentation, other metabolic activities get activated by the yeast, and it takes away from some of the ethanol production. Also for the 28 Brix condition, again, we would expect to see that all the wines made from the 28 Brix would be in higher ethanol concentration than our control wine, which was just at 23 Brix, and certainly that was the case. Again, the on-vine fruit not getting up to quite the level of ethanol as we had expected. Keeping in mind, all of these wines went to dryness, okay, so this was a little bit of not what we were expecting to see. Sorry. In terms of acetaldehyde concentration, again, we do see that in the starting fruit, we have a little more acetaldehyde starting out in the kiln fruit, okay? But by the time we get to the wine, it didn't really differ too much in the final wines. And we see here that the barn actually produced the highest levels of acetaldehyde. Acetaldehyde is that compound that people describe differently. Many associate it with sherries because it's produced in high quantities in sherries based on that style of wine production. Others might describe it, if you bit into an apple and let the apple brown and then smelled it, you would start to get some acetaldehyde coming off. So these are some of the characteristics. Tends to be sort of a sweet smelling compound coming off. So similarly, at the 28 Brix, we're seeing a bit higher level of acetaldehyde being formed using fruit coming from the traditional barn drying technique. Acetic acid, another one of those compounds that we were a bit concerned about. Remember, in the fruit, as it's drying, is it going to build up too much in the fruit, and is that going to be a problem in the final wine? And again, where we saw it building up in the initial fruit here in the kiln, we really didn't see that translating excessively in the final wines. Again, the barn tended to have higher levels of acetic acid building up. Now, I say higher levels. I also want to draw your attention to what the values of these acetic acid levels are based on the scale. They're only going up between 0.4 and 0.5 grams per liter acetic acid. That's not considered a high level of volatile acidity for these wines. Now, mind you, these wines have been stopped after primary fermentation, so they haven't gone through barrel aging where there may be a little further increase in VA that can result in the wines. But overall, we aren't producing excessive amounts of acetic acid. What I was happy to see is that even though we're building up a little bit of acetic acid in the fruit of some of our treatments, it's not consistently translating in the wines at 26 Brix. It's a bit different story once we get up to 28 Brix. So yes, the kiln fruit did show a higher level of acetic acid showing up in the 28 Brix wines that resulted. 28 Brix is a slightly different dynamic for the yeast to ferment compared to 26 Brix. It starts to evoke some of the osmotic stress responses in a much larger way than what we see in the 26 Brix fermentation. When yeast get stressed by high concentrations of sugar, their natural response is to produce higher levels of glycerol. It helps them deal with this stress. Otherwise, they lose all the water from the cells, they shrink, and they die. So they have to produce glycerol to combat that stress. But when they produce glycerol, it goes hand in hand with producing higher levels of acetic acid. So you'll also see in all of these 28 Brix wines, they produce higher levels of acetic acid than what was found in the 26 Brix wine, and that has more to do with the osmotic stress that the yeast are under. So why we're spending some time looking at things like acetic acid and acetaldehyde in the initial fruit compared to the wines is that we know as that fruit gets further concentrated and we get more sugars developing and putting osmotic stress on the yeast, there is going to be a higher propensity for the yeast to produce things like acetaldehyde, acetic acid, ethyl acetate. So we don't want too much building up initially in the fruit because we know we're going to get some building up in the wine during this style of wine production. Ethyl acetate, again, nothing too concerning here with the ethyl acetate. The levels are between 40 and 50 milligrams per liter. We really don't get too concerned about that until it's 100, 150 milligrams per liter. So that was a good news story. We're not building up high levels of ethyl acetate. Similarly with glycerol, I mentioned earlier, we get more glycerol produced in wines because of the osmotic stress the yeast are under at the higher Brix, and so they'll produce more in the 26 Brix versus the 23, and they'll produce even more in the 28 Brix. Glycerol isn't going to take away at all from the quality of the wine. It's just an indicator of the stress that the yeast are under during the fermentation. Okay, I wanted to move on to some of the cost analysis associated with these wine styles. So one of my former students in the lab, Ian Bach, carried out a bit of an economic analysis for these wines to look at comparing the grapes that are required to produce these wines relative to the control. Okay, so knowing that we're going to lose some volume of grape, right, because we're drying them, but we're still trying to produce a similar volume of must to ferment into wine. So how much more grape tonnage would we need compared to the control if we're going through these different ways to dry the grapes? Okay, so I just wanted to walk you through this data first from 2011 and then from 2012. So we've got fruit at 26 Brix and then fruit at 28 Brix. What we can see, not so surprising, on wine, where we have higher losses due to wildlife predation and breakdown of the fruit, it'll take 73% more fruit to produce that same volume of must compared to the control. Okay? That's not so surprising. Maybe the numbers are a bit surprising. We didn't think we were losing that much, but there's the comparison. For the kiln, okay, it only takes 12% more fruit to reach that same volume of must compared to the control. For the barn, it was about 30% more. For the drying chamber, about 23% more. Okay. So these numbers are mainly in line with just the weight loss. The barn in 2011 had a little bit of fruit breakdown from Botrytis, so we had to get rid of some of that fruit, so that added a bit to the cost. And the greenhouse in 2011, I don't want to put too much weight on that because that was the experimental year. We had the tarps on. We actually threw out about a third of the grapes, so there was a reason for that. So again, we've got the amount of grapes that it took to produce our volume of wine for the control. And then we compared the grapes required to get that same volume of must compared to the control from each of our drying treatments. And it separated out 26 Brix versus 28 Brix. So again, on vine, okay, we have the highest losses. In 2012, we actually, later on in the season, as the grapes were going up to 28 Brix, we had more bird pressure and loss of grapes to wildlife predation. So for 26 Brix, we required 68% more of the fruit. By 28 Brix, wow, you know, it took a lot more fruit to be able to get that same must volume. In terms of the kiln, very, very low losses, okay? Basically, this is just the loss of weight from dehydration. Okay. For the barn, also fairly low, similar to the kiln at the 26 Brix. And the greenhouse, we feel this is a fairly good estimation of what was going on in the greenhouse. So it took about 24% more of the grapes to reach that same must volume. We did have a raccoon unfortunately, that got into the greenhouse and loved Appassimento grapes. So they ate a little more of the tonnage by the time we got to 28 bricks. So again, research doesn't always go as planned. So the numbers for 28 bricks are a little bit higher for the greenhouse because of that additional wildlife loss. So we're going to continue with this economic analysis because what we haven't incorporated yet are final wine volumes because some of these numbers are going to change a little bit because with the lees that results from the different winemaking techniques, we actually have different volumes that we finally actually rack off for the wines. So numbers are slightly different when we actually look at wine volumes. Then what we haven't incorporated yet are some of the costs of the different techniques. So we haven't incorporated, for instance, the cost of the drying chamber in 2011, the input costs, the energy costs. We haven't included that for the kiln. So we hope to have a more extensive economic analysis as we continue through the project, so it becomes useful information for our various industry partners as they're looking at investing in the different techniques for drying the grapes. Did you have a question?
Well, I was wondering for the greenhouse at 26 bricks, was the raccoon involved as well?
Just for maybe 4 clusters of grapes. It would have a small impact on that number, but we weren't making the connection that the couple of stems that we saw beside the tray were due to a raccoon until the following week when it was a couple of trays.
24%
No, I don't have an explanation on that, and it's the very first year that we've done the greenhouse, so I think we need to replicate this again and get some comparative numbers. The previous year just wasn't a good assessment because of the different way that we ran the experiment.
Did you find any botrytis in the greenhouse at all?
So kudos to Jamie. His fruit was so clean this year that there just really wasn't any botrytis to speak of in the fruit, which impacted an additional study that we wanted to do last year. This is just the same data that compiles 2011 and 2012 data. So all of this will be posted on our website, and you'll have time to look at this data in a little more detail. So in terms of this part of the project, the sensory evaluation of the wines was carried out in the fall through December, was completed by December, but the researcher in charge of that, my colleague Gary Pickering, is working up the data, so I'll have to present the sensory data to you at a later date.
Gary, can I assume that you won't have sensory data for the drying room?
For the drying chamber for 2012, not at all, because we don't have... we didn't have the fruit, but for 2011 we do have those wines.
Oh, so you have, okay.
Yeah.
You mean wines in 2011?
Yeah,
right.
okay. But not for 2012.
What happened to it? What happened?
You guys did the experiment separately. So Vineland is doing that project.
They cut you off?
Yeah, there was a commitment to enter the project, if I understand correctly, in 2012, and unfortunately the commitment wasn't filled, and they weren't able to do comparable data for 2012. So it would be nice to see if it could be included in 2013.
Yeah. But we can talk about that maybe afterwards. Certain. So we're still compiling the rest of the chemical data for 2012 and the microbial analysis. So again, I don't have all that. It's a lot of data to present to you. I did want to just take a couple of minutes, if you've got time, to share with you a little side project that we initiated in 2012. Didn't end up being successful because Jamie Slingerland did such a great job out in the vineyard. The fruit was so clean, we just could not get botrytis induced in this fruit. But there's an increase in looking at the impact that botrytis has on final wine quality for this Appassimento style of wine production. So a big question is, should we always discard the botrytis-affected fruit, or is it important to have a certain amount of botrytis and the right type of botrytis infection in the fruit to add complexity to this wine? So recent research that's coming out of northern Italy from the research groups at the universities and research centers around Verona, Italy, is pointing to the role of some level of contribution from botrytis, but in the noble rot form, not in the fuzzy gray rot form, and I'm going to show you a picture of that in a minute. So we thought in 2012 we had some extra fruit available. We thought we would set up an experiment in the barn to actually try to look at different levels of botrytis infection in the wines, allow some botrytis to develop. We thought it would have the greatest chance of success in the barn because it's a longer drying period. And we wanted to try to target infection levels at 10 or 20% botrytis compared to clean fruit and see what the impact was on the flavor profile of the wine. So in order to do that, we needed to separate out the different forms of botrytis for this experiment. So this is these are pictures from the research group in Italy of a berry without any botrytis, a berry with noble rot form of botrytis, where the botrytis is inside the fruit, and it's hard to see here, sorry, but that fruit has a bit of a red tinge to it, okay, compared to the blue-black color of the intact berry. What they want to avoid is the gray rot form, okay, the fuzzy form. So it's when we're talking about 10 to 20% botrytis infection, we're talking about this noble rot form of botrytis, where the berries are desiccating. Botrytis is internal to the berry. It's breaking down the skin structure of the berry, and it's turning the berry a little bit of a reddish tinge to it. So in order to set up a similar type of experiment, we took berries from our trays and tried to separate them out into these various categories. We had black withered, so they're starting to shrink up a little bit. These ones had a little bit of a red tinge that really you could only see under a bright light, so it's not depicted very well in this picture. Red withered, and then a positive sporulating control. And my goodness, how hard was it to find 5 berries out of this fruit that were sporulating as positive controls. We were on a hunt. The fruit was that clean this year. So taking those 5 different categories of fruit, we then wanted to mash them up, plate them out, and see if there was any botrytis present. And for those 5 categories, the only category that showed botrytis was the positive control, the one with the gray fuzziness on it. We then took those 5 categories of berries, okay, put them into an incubation chamber with lots of humidity and warm temperature and tried to induce botrytis. And when we tried to do that, again, after 7 days in conditions that normally would allow for botrytis to develop immediately, we couldn't get any botrytis developing except on those positive control berries. So best laid plans were foiled this year, but we have developed the protocols that we want to look at. Obviously, what we thought was red and maybe had a bit of botrytis starting in the fruit ended up not being the case. They were just slightly underripe berries. But again, the idea is that we'll develop some type of visual clue to indicate different levels of botrytis infection and then try to target 10 or 20% infection and compare what the wines look like. Yes?
So what is the sporulating berries that be considered sour rot?
Yes, that's typically that would occur earlier on. Well, no, I shouldn't say sour rot, but it may be part of a sour rot complex, but it's referred to as gray rot or gray mold, but often it's undesirable, yes. Okay. So with that botrytis experiment, we tried it with 3 different trays in November, in December, and January. Each tray was basically the same story. We just could not get botrytis induced. So we'll try again next year with this and look at maybe talking with our industry partners to see if we happen to have really clean fruit again next year, do we want to maybe spray a Botrytis inoculum on the fruit to try to induce this, as opposed to just allowing for the natural Botrytis to develop? Additional questions we need to look for in 2013. What variety will we focus in on? Will we repeat Cab Franc again? Will we do any other modifications to the various drying chambers? Maybe looking at different temperature regimes within them. We also have a Brock isolate yeast that we're further characterizing and looking at its use specifically for Appassimento wine production. And why we're excited about this particular natural yeast isolate is that it's a very low producer of acetic acid, ethyl acetate, acetaldehyde. So again, with this style of wine, which has a tendency to produce higher levels of those compounds, this may be a unique yeast focused for Ontario wine production. And we'll continue with the cost analysis. So overall, summarizing, we think that this Appassimento wine style, this process shows great promise for Ontario, and definitely in our experience so far, great promise for full-bodied red wines. The drying process does impact the wine flavor. The wines developed from the different drying techniques all have some unique characteristics associated with them. So it'll be, depending on the style of wine that you're trying to develop, you can match that style of wine with the type of drying technique that you're looking at. We're looking at repurposing some of these drying facilities from other industries. Definitely there's tobacco kilns that are available for this, but also in this area in the fall time there's open access greenhouse space available, and this may be another avenue for growers and wineries to take advantage of. We feel this is a new innovative wine style for Ontario that has the benefit not only of overcoming some of the climatic barriers that we have fully ripening our fruit year in and year out, but also developing a new signature wine style for the region that will help grow our industry. So with that, I'd be remiss if I didn't acknowledge all of our partners, and again, you can imagine on a project this size, the number of partners. We have our colleagues at Vineland Research and Innovation Center, Michael Brownbridge, Bernard Goyette, and Jean-Beau Liu. And Bernard and Jean-Beau are focusing in on that drying chamber. Michael's technician, Irina, who does the mold analysis, and then the whole harvest team that's focused at the Cherry Avenue Vineyard that helps us with the harvest time. At harvest time, Terence Van Rooyen from Niagara College and his staff and students that are part of this program. My colleagues here at CCOVI, Gary Pickering, Vincenzo DeLuca, Jim Woolworth, and myself that work on this project. Lisa Dowling, the technician in my lab that did all of that berry analysis, sampling each day, each week for all the sampling times for all of those different drying processes. We have our CCOVI harvest team. We have additional technicians. I see Linda Van Zuyden in the back that carried out a large quantity of that chemical analysis. Fred DeProfio for winemaking. Ian Bach for some of the environmental and economic analysis and for setting up that initial Botrytis trial. And Christina Huber, who also did a lot of our microbial identification here. As you can see, we also have a number of industry partners, and Jamie Slingerland from Pilotary Estates is here, Cave Spring Cellar, Rife Estate Winery, European Planners, Sunrise Greenhouses, Integra, Graham Rennie, who's in the audience, Angels Gate Winery, I guess, would also be included in as your partner, the Grape Growers of Ontario and Ontario Grape and Wine Research Inc. that funded this, as well as both the provincial and the federal government funding agencies that allowed us to bring this project forward. So with that, I'm happy to entertain and answer any additional questions from the audience. So, yes, certainly.
I noticed at the beginning, we're just referring to the acid reduction, so strictly due to the grape metabolism,
yeah.
Of malic acid?
That's what we're surmising. I can't say that for sure because there are microbial populations, present on the grape, but definitely grapes can undergo a process of metabolizing components. They are biochemically still active, these grapes, even though they're cut off the vine. So,
if they're ripening, where
yes.
the malic acid goes down.
Yes.
The tartaric acid was unchanged?
We didn't measure tartaric acid specifically. That tends to be a more stable grape acid and likely didn't decrease to the same level. We do have measurements of overall titratable acidity. I didn't present all of the chemical analysis here today, but we feel that the majority of the drop in the TA is due to the malic acid degradation.
Was the final TA compared to the control comparable?
Yes. So again, what we're getting in the initial TAs of the juice in the initial mush, it depends on the year, between 5 to 7 grams per liter titratable acidity, and then we're not further increasing the concentration of those acids, which is what you would expect if you were just losing water. So they're staying the same or slightly dropping. Yes. Okay.
Can I just get you to repeat the questions over and online?
Oh, yes, sorry. So that initial question was about malic acid drop and the overall impact on titratable acidity. Jamie, yes.
Okay, seeds. Quite often we measure sugars,
yes.
But it's seed development as well. And when you harvest, were the seeds dark brown? Was there any tinge of green to them? And I guess Oh, they were full brown.
They were ripe, yes.
I guess the question that to be asked is, at some point in time, is the drying methods affect those seed colors?
Yes. And we haven't, good question. We haven't included that in our visual observations, but maybe that's something we can look at down the road. What we are measuring, which I did not present today, is a full phenolic profile in the grapes during the drying process as well as in the final wines. Okay. So that we're gaining information on, are there differences in that overall phenolic profile, not only in the fruit, but what gets extracted through the winemaking process.
The reason why I asked the question, there's been discussions that if green seeds or partially green seeds are able to turn brown during that drying process, that they'll get rid of some of those greener tones.
Right.
And what is going to happen to seeds that are completely green?
Right.
And the thought process is that they won't turn brown in that drying process.
Now, with how we've set up this experiment, we're taking fruit that's ripe to begin with. I mean, it's 23 bricks. I believe in 2012 our TA was around 5.5 grams per liter. I think in 2011 it was closer to 7, if memory serves me correct. So our fruit was ripe. In 2012 it was ripe. It was, not on the green side. Could that be another iteration that we look at, in future years, taking fruit at 21 bricks versus 23 bricks, and doing a seed comparison and some of the phenolic profile comparisons? I think that's something we could look at. It's not something that was built into the first 2 years, but I think useful information in terms of, can you use this technique? To further ripen the fruit off vine in a year where, we're just not, they're not moving past, that 21 bricks and they're green and, really high acidity. I don't have the answers to you on that. I have a feeling that there's going to be limitations, of what these techniques can do for the fruit. The approach we took was to use good sound fruit right from the start for this project, and then further enhance that fruit structure through the drying process.
I was wondering with the kiln experiment we did, especially with that little mistake at 40 degrees.
Yes.
So perhaps if the grapes would be dried at 40 degrees, you would not have all that increasing amount of acidity.
No, we actually I think it's going to be the opposite. I think at the higher temperature for drying, bacteria like to grow at warmer temperatures. 37 is kind of their magic temperature. So that their preferred temperature for growing. So I think that, although I can't say this for sure, it's something we're looking at, the microbial population on the fruit prior to drying and after drying, and we're going to compare the kiln fruit to one of the other techniques that was at the cooler temperature to look at if there was a difference in the bacterial populations. But I would hypothesize that the warmer the temperature, the higher the buildup we're going to get. Yes.
So in all the cases, were the grapes stored in or placed in the same type of trays?
Yes.
Spacing between trays?
Yeah, not exactly the same, but fairly similar. So let me just go back to that picture.
That was a few minutes ago.
Okay. So these trays that are used in the barn are the same trays that we used in the greenhouse. These trays in the kiln are very similar to what we used in the drying chamber. Okay, so we're making use of the trays that are used commercially at each of the various operations. So these trays are more square bread trays, bit thicker plastic. These trays are used for post-harvest production fruit for various storage techniques. So they're actually designed specifically for post-harvest storage of fruit. There's lots of airflow in these. So we haven't used the exact same trays through each of them, nor do we have the same layout or dynamic. Keep in mind for this picture here, this is a picture within the barn at Cave Spring that has also their commercial production. We're only doing 250 kilograms of fruit. So we're doing a much smaller scale version. So for us, 250 kilograms of fruit is this much fruit. Okay, that's in the kiln.
So, Debbie,
yes.
Didn't see it in the economic model, but one thing that I found with the chamber is that we have much higher volume to surface ratio. We really pack it in, you know, so for a square foot you got a lot more grape drying. And I just wondered if you know any sort of comparison.
We don't have that done yet. We haven't carried through our analysis that far yet. I think that's a good point. I think so this is a picture of the drying chamber. I think commercially in the kiln, they're packed quite tight. It would be quite similar volume, like square footage wise, to what we see in the chamber. In terms of the barn, I think there's a little more space in between the various rows of the grapes, but again, the barn is packed fairly tight. We'd have to go through and actually, do those calculations to relay the info. In the greenhouse, this was our first year actually using the greenhouse as a real greenhouse. So we just did one line of the fruit trays on the table. But again, this could be stacked up to the height that we could do safely without having the trays tip over in the greenhouse. So we could go several high in that model. Yes.
I guess they're plastic. The trays are plastic.
Yes.
Holes, I presume, aeration.
Yes.
Aeration is possible.
Yes.
I was wondering what would make a difference as far as there's a breakage and a barrier that would tend to absorb the juice kind of thing. Yeah, I read Tuscany or
yes.
part of it, especially actually the shins on time. They just lay them on straw, supposedly. I guess that's really the real old.
Very old technique. Typically, they're using similar type of tray setups.
Strictly one layer.
Yeah.
The grapes shouldn't be.
Yes. So in all of this, that's a good point. You'll see that we're only putting one layer. Maybe you can't see that very well, but we're only putting one layer high of the fruit, so that we don't have cluster on top of cluster that could crush the bottom clusters. What we're trying to prevent is any damage to that fruit, to the berries, so that it's drawing intact.
In the average operations, my son spent a month in Verona harvesting grapes, sacking them, and they were doing them exactly to, if you look at the drying chamber, that's what's happening commercially as well in the kiln. They're stacked up like that. If you go to Rome, Italy, you'll see them almost identical, except that they'll have 1, 2, and 3 skids on top of each other. They'll have 52-inch fans. They'll have a wall of them. And so exactly the same containers, exactly the same type configurations, and air going through them, and exactly the same one layer of grapes on each of the trays.
Yes.
But Jamie, they don't use kilns. You don't use a kiln there.
No, they do have.
I mean, they don't use a heated environment.
Massa does.
But not to 40 degrees or 30.
No, not 40 degrees.
Because the question I had is, you know, if you read the Massa literature, they've got a decade's worth of scientific research on this. They're including their most recent, they do the annual in Italy presentation. Right? So the most recent one they had, they continue to reinforce the notion that the reason that wines are so good, besides the varietals they use, is that it's an incredibly long, slow drying process where you get all of these changes occurring in a grape that you can't get in a short period.
So this is the point of this project, Graham, to actually ourselves do those measurements where we purposefully have the short dry time, we have the long dry time, we have intermediate dry times, and we can compare, all with the same grapes, you know, that go into these different methods. How different are the wines? How different are they? So our analysis will be descriptive analysis. What are the different profiles that are coming out of the wine? Not what's better or worse. In my opinion, they're all great wines. They're all sound wines. But can you emphasize certain qualities using one technique versus another technique? Then I think what also comes into play from a winery's perspective is, what are you trying to achieve with this? You know, is a kiln model a better economic model for you because your grapes go in and within a week they're coming out and you're fermenting? Or do you have the space and ability to store these grapes for, you know, 1 month, 2 month time period, allow that slow dry process, and what's the added benefit that you're getting at the end of the day in the wines that are produced? I think for us right now, we're at the very early stages of this, and we don't have all of those answers just yet, but we're using different grape varieties than what is being used in Italy, and we're also trying to look at developing our own signature style, a unique Ontario style that isn't, you know, an Amarone imitator. Right.
So the sensory testing is going to be critical.
Yes.
Which means you have to get drying chamber grapes.
Yeah, and we do in 2000.
You know, only have one test, right?
Yes.
And you need more.
More, yeah.
Okay, we can make that happen.
Would it be fair to assert that there could be a number of variables between a number of the methods that could be advantageous? For example, on the vine, maybe a producer in the right vintage leaves them on the vine until they're 24 bricks, 25, and moves them to a drying chamber for a shorter period of time, or to a kiln, but then maybe alternate the kiln. Why does the kiln have to be 40 or 30 Celsius? Maybe 25 Celsius, maybe 20 Celsius is that magic number. And therefore, you know, utilizing some of the advantages of each one to find that happy medium.
I think we're only limited by our own imagination, and I hope what our research project is going to bring forward to us is useful information that helps define what happens in each of these techniques, so that the winemakers, winery owners, vineyard managers can make those educated decisions based on knowing what happens in each of these processes. So we have 2 more years of funding still to continue with this project, and many, many more questions to answer. So I thank you very much for joining me today for the talk. And I would like to acknowledge that on Tuesday next week, we have an additional lecture, part of our CCOVI Lecture Series. It's Ron Jackson. He's one of our CCOVI Fellows. Many of you may know him as the author of Wine Science. And he's going to be talking to us about wine language, insight into the mind of the taster. So we're very much looking forward to that talk Tuesday. Then we'll have our regular CCOVI lecture on Wednesday again, and that will be Dr. Jeff Stewart talking about bioactive polyphenols in grapes and wine. Thank you very much.